So it's not necessarily purely something that gives us a sense of what modeling choices to make. It's something which may be of interest to the broader community in terms of summarizing impact. Okay, so what I'd like you to do is reopen this model. It's in the sample models or example models in AnyLogic, SIR Agent Based Calibration. This is the model we saw last time when we were looking at the Monte Carlo 2D histogram and the issues of variability, stochastics, within the model. And I'd like you, once you open it, to click on the Monte Carlo 2D histogram experiment within that model. So I'll go see if I can fire it up here. I think I already have it loaded. Yes. Its complaint gives it away. There it is. And I was futzing around when I was trying to decide what slides to put in here. So here it is. It's SIR Agent Based Calibration. And if you go double click on this guy here, you'll see this this pointillist diagram. Does anyone remember what this is? What is this structure that we see here? Yeah, it's a Monte Carlo 2D histogram. So it's a chart actually that displays data from a histogram 2D data set. And each of these, you'll recall, each of these dots, which here are thrown down in a sort of modernist interpretation, each of these dots is a cell, or each of these sort of little boxes is a cell, which summarizes the number of trajectories that fell for this small interval of time, where the values of those trajectories for a given quantity of interest fell between sort of this level of value along this axis. So if this was number of infected people, this would suggest there's a certain number of trajectories. It's darker, so probably quite a few, whose value at that interval of time, say mean value at that interval of time, fell between, maybe it's about 900 and 1,000 people who were infected. Okay, so we saw that last time, and we're going to build on that today to help understand the impact or to interpret sensitivity analyses. Okay. So when you double click on this, what you'll notice is that the initial statements of parameters for that histogram, and I've got to, I was preparing these slides, so I've got to go back and restore these, I think, to their original value. But it should be fixed. I think initially it should look like this for you. In other words, it specifies for this experiment that we're going to use fixed values for all these parameters. And we last time had something along this lines. Why did we do this? Does anyone remember? Why would we have been looking at variability in results if we had all sort of fixed values for these expressions? It was because we were looking at variability due to what? Yeah, it was underlying stochastic. So given the same values of those parameters, you still have variability. And we saw that it was modest, but could make the difference in some cases between an infection that never took off and one that did. In other words, one that just petered out after 1 or 2 or 3 people getting infected versus one that took off. Here, we're going to be looking at something different. We're going to be looking at varying these things. And what we can see specifically is that for each one, we can pick whether it's a fixed, so I selected varied in range here. And then given that, we can either select a parameter value as fixed. We're not going to be varying that. Or we could specify it as varying in a range according to some systematic variation. And there's a small set of limited options available. So I'd like you to select, for the very first of those, average illness duration, select range. And I'd like you to select a minimum value of 5, a maximum value of 5, in a step of 5, okay? So that yields 5 different possible values for this, 5, 10, 15, 20, 25. It's going to systematically examine the results varying that parameter, starting at 5, successively incrementing it, 10, 15, 20, etc., with this step of 5. Right? So this is a so-called range variability. And you'll notice here that when we selected this varied in range, that we did so, and when we did so, this number of runs thing, which had been visible before, is now grayed out. It's now disabled, in the words of a UI. So I'll try to just highlight that now. When we had this, remember we set the number of runs? And actually, AnyLogic's little information on it said 100 runs, and we pointed out it's actually 200. So that is something that we can set here quite explicitly. When it's varied in range, we don't have that option. Instead, it's implicit. It's implicit in the variability we exhibit, or that we're requesting, the ranges we're requesting. So let's run that thing. So let's run it, and we will sort of see what results from that variability. So it's 700 replications, but that's not relevant anymore. How many replications do you think it's going to run? How many simulations is it going to do here? Well, does it look like a large number or a small number? Small. And I'd say it did 5. Where did this 5 come from? Yeah, exactly. It's just 5, 10, 15, 20, 25. So this is what I'm saying about the number of runs being implicit or derived from, implicitly specified by the ranges you specify. Okay? Yes?
Can you do 24 runs?
Yes, you can. So you'll recall that that last time I argued that there's AnyLogic uses, in a somewhat non-standard terminology, a threefold characterization of the hierarchy of runs to be made. So there was, on the one hand, an experiment. On the next hand down, there was a simulation. The next hand down, they have what they call a replication. And I said replication is more commonly used, at least in my neck of the woods. We use the term realization, a realization of an ensemble. So it's a particular run of the model with a specific random number seed, particular sort of stochastics associated with it. It's a draw, as it were, from a distribution associated with the stochastics. And here, what we're examining is a situation where we're running 5 simulations, each of which is composed only of one replication. And because AnyLogic articulates this division, it is possible to have multiple replications per simulation. Okay? So in other words, and I think this is a very perceptive thing Deval is asking about, because what we see here is a set of trajectories, but the clear question to ask is, well, how much of this variability that we see here is due, on the one hand, to the parameter varying, versus, on the other hand, to the underlying stochastics, right? How do we know that if we ran one of these 5 times, we wouldn't see a similar measure of variability? Maybe the variability we see in the parameters just washed out by the variability in stochastics, right? So that would be a good way to articulate that. Of course, there's other ways of investigating. I mean, you could run 5, realizations for a given single one of these and see the measure of variability. In fact, let's do that just so we get the heck, we can exercise a little bit. So we'll do it with 5 here. It's on a value of 15, and we'll run it. Sure, we'll run it 200 times, right? This is what we really did last time. And we saw some measure of variability around it. So it was filling in. And indeed, there's different trajectories being filled in. But broadly speaking, they're clustered in a way that those others weren't. What we saw is kind of a progression that was systematic there. a progression was in fact dominantly shaped by the variability in the variation of those parameters, variability of the parameters, rather than variability due to stochastics. So we could run this out till 200, or indeed till the cows come home, and what we'd see is that it's actually reasonably tight in terms of the variability. Yeah.
Sorry, you specified about replications, but
Right. So there's a. I'm going to be talking about that issue in our calibration calibration time. But to say that we'll see there where, so if you go in and you look at, and I believe it's in advanced here. So there's this before simulation run and after simulation run component here, and we'll see basically how you can use that to shape the number of realizations. Okay, I'm not going to address it right now. I don't have the slides for that and so on. So we're going to have to come back to that, but I'll make sure that that's answered within our calibration one. Okay, because it's a major point. Because in calibration, as you can imagine, where we're trying to match the model output to some datum, we want to understand how the model output is variable for a given set of parameters, how it varies, so that we are matching up indeed against for the best parameters, not for just the best chance of stochastics. Anyway, so we'll come back to that, but the parameters here are varied within the range, and we can do a range this one. We'll put it back. You'll notice it remembers the values we put there. Now what I'd like you to do, so that's a one-way sensitivity analysis. For multi-way, we're going to examine the results of changing multiple variables at